Hello everyone. A very good evening to all of you. So, can you please confirm is the sound is clear? Screen is visible? Yes. So, I hope so you might have enjoyed the first session of CSIR NET question discussion yesterday. You might have got a greater insight on where to focus on and where you are confident on, right? Yes, can you please confirm in the chat? Yes, very good evening, Safalta. Okay, so can we start with our session today? Yes? If so, please type BIO in the chat box. Yes? Okay. Okay. So today we are going to discuss about some questions related to Unit 2, that is cellular organization. So I'll be taking the questions from Unit 2A, 2B and 2C. So remaining questions will be covered by Dr. Violet. Okay. So let's begin with our first question. And the first question is on your screen. So this was from shift 1. So first I'll be covering shift 1 questions and then I will switch over to shift 2. In eukaryotic cells, covalently attached lipids help to anchor some water-soluble proteins to the plasma membrane. One group of cytosolic proteins are anchored to the cytosolic phase of membrane by fatty acyl group. So they have mentioned example myristate or palmitate. These groups are generally covalently attached to which amino acids that is present at the N terminus of polypeptide chain. So here are the options for you: glycine, tyrosine, serine, and lysine. This is a very easy and a direct question. So let me see how many of you are able to give the answer. Yes? What is the answer for this question? If you remember a diagram, a picture from Leninger text, you will be easily able to grasp this question. Yes, I did not see any answers from your side. Still have confusion with this question? Okay, I got an answer from Ashwini. You have opted for 2. What about others? See, they are asking about lipid anchored proteins over here. So you have got different types of lipid anchored proteins like palmitate is there, myristate is there, then farnesyl is there, GPI anchored proteins are there. So how they are connected? In which terminus they are connected to which amino acid? Yes? I can only see one answer coming up. What about others? You have not attended it? Okay, let me show you the answer. The answer to this question is 1. Now, why exactly 1 has come? See, in the question, they had asked one group of cytosolic proteins are anchored to cytosolic phase of a membrane. And here they had confused you by saying myristate or palmitate. So it could be either of this thing. But if you read the second part of the question, what you will find? They have asked for the amino acid present at the N-terminus region. Right? So they have already given you the answer in the second question, second part of the question. So easily, if you will remember this picture, you'll be able to get the answer. So the answer will be glycine. If you look over palmitoyl group, they are also attached to the membrane, but they are attached to the cysteine residues. And they are not attached either to the N-terminal region or towards the C-terminal region. Rather, they are attached to the middle point. Similarly, if you look into the farnesyl or geranyl group, Where they are attached? They are attached to the C-terminal region. To which amino acid? Cysteine residues. Similarly, if you will find GPI anchored proteins are there. Where they are attached to C-terminus region. So if you simply remember this easy diagram from your text, it will be easy for you to fetch the marks. Right? Don't you think this is an easy question? Now, after looking into this picture, fine. So let's move on to the second question. Again, a very direct and very easy question. The movement of proteins bigger than 50 kilodalton across the nuclear envelope requires what? Sec61, Tom 20, Tim 44, importin beta. What will be the answer here? Now, I guess everyone might have attended this question. This is that much easy question it was. Yes? What is the answer for this question? Now, I can't believe that you have not attended this question. Any protein which has to be imported within the nucleus should have a common protein for them. What is that protein name? Yes? Yes? Very less response. Can I get the answer for this question? What is the protein that is responsible for the movement? See, whichever protein you consider, if it has to enter inside the nucleus, what will be that protein? Yes, very good, Madhura. Very good response from your side. What about others? Yes, Ashwini, Safalta. What are your answers? Yes, Nancy. Very good. I can see all are giving the correct answers. Yes. So the correct answer over here is importin beta. Yes, very good, Devanshi. Now, if this is the correct option, why do you consider the other three options as incorrect? See, you can easily eliminate these two options. Why? Because this is the translocon present in the outer mitochondrial membrane. And this is the translocon that is present in the inner mitochondrial membrane. Now coming to Sec61. Where is Sec61 present? These are the translocon that is present within your endoplasmic reticulum, if you remember. Right? So without eliminating also, you can get the answer for sure. But just for an explanation, I just explained you what are the meanings of other options. Okay. Yes, Anu. Yes, Akriti. All of you has given the correct answer. So the transport of proteins around 20 to 60 kilodalton will be taking place through your NPC, that is nuclear pore complex. So this is a highly energy requiring process. So definitely there will be a hydrolysis of ATP or GTP will be taking place. So the best characterized protein import pathway is the classical NLS dependent pathway with importin alpha and beta. So these are the receptors which carries the protein inside the nucleus that carries the substrate towards your NPC. Right? So this is the correct answer. Now just to remind you, today Shaurya batch is going to be started off and the admissions are getting close sooner. So make a wise decision. Once you are into Shaurya batch, you will be able to access all the online classes. You will be able to interact with our lecturers. You will have access to 24/7 doubt solving sessions. In fact, you will get the printed hard copy materials as well. So if you want to ask anything, please call to us in our toll-free number 1800-1200-1818, or you can reach us by 080-5099-7000. Okay. So moving on to the next question from shift 1. So these are few statements with respect to nuclear transport. See, nuclear transport is given very much priority when it came to shift 1. You got a question in the part B as well as in part C as well. Now, these are the few statements given. You have to find out the correct combination of statements. First statement, RAN GTP levels are higher in the nucleus compared to cytoplasm. Nuclear import receptors, that is your alpha and beta, will be shuttling between the nucleus and cytoplasm. NTF2 will transport RAN GDP into the cytosol. Export of mRNA is not directly dependent on RAN. tRNA and microRNA export is mediated by exportins. Now, can you able to find out the correct combination of statements from here? Yes, find it out. Everyone, come on, everyone join. Everyone try to give the answer. See, you are giving the right answer, wrong answer hardly matters. You are learning something new every time. So if you are right, you will be confident. And if you are wrong, you are learning something new. So don't obstruct yourself. Give answers, no matter what it is. So what will be the correct combination of statements here? So you have idea about RAN GTPs, where they will be present. You have the idea about your nuclear import receptors. Then NTF2, nuclear transport factors. What are the exportins that are present for tRNA, microRNA? Yes? Any idea? Yes, I can see Safalta Madhura has given the answer. What about others? Yes, what about others? You still have time. And this is also a very easy question, not so that much tough comparatively. If you know, you will definitely able to give the answer. Yes, Akriti, Devanshi, what will be your answers? Yes? See, the correct combination of statements or the correct answers will be here, according to CSIR answer key, it is 3. Now, why it is 3? 3 includes A, B, D and E only. Yes, Kavita, Devanshi, Akriti, I can see your answers. See, why C is only excluded from here and why all are included? Let's find out. So first what it is mentioned, RAN GTP levels are higher in the nucleus rather than the cytoplasm. So let's have a look into the picture over here. See, RAN GTP. RAN GTP, how they are formed? RAN GTPs are formed with the help of RAN GEF. And where exactly the RAN GEF will be located? RAN GEF will be located within the chromatin. Chromatin is present where? Within the nucleus. So obviously, your RAN GTP will be present in higher level within the nucleus. Corresponding to that, you are having RAN gap, right? Which is responsible for the hydrolysis. So this RAN gap is present within the cytosol. Upon hydrolysis, what you will get? You will get the inactive form of your RAN protein, that is RAN GDP. So they are present where? They are present within the cytosol. So remember, RAN GEF present within the nucleus, RAN gap present within the cytosol. Now coming to the exportins, meaning the way tRNA and mRNAs are exported. As you can see, the proteins that are responsible for both import and export inside and outside the nucleus are known as karyopherins. Okay, so from here you will find out that exportin T and exportin 5, these are the two proteins responsible for the transportation of tRNA and the microRNAs respectively. Correct? So this is the proof for the other statement. Then As you can see in this picture, how exactly the proteins are getting imported in the nucleus. The red color one which you can see is your protein that contains NLS signal. NLS signal in the sense nuclear localization signal. And this is being carried out by whom? Importin alpha and importin beta. So what are this importin alpha and beta are? They are considered to be the receptors for this protein. So this receptors and the protein will be combined together and they will tethered within the NPC. NPC means nuclear pore complex. So with the help of cytoskeletal protein, the entire complex will be taken up inside. And via nuclear pore, the protein will be taken to the nucleoplasm. So now what happened? Your NLS containing protein has reached its destination, that is your nucleoplasm. Now, what is the job of receptor? The job of the receptor is to carry the protein to the nucleoplasm and then recycle back to the cytoplasm. So for that, you are having RAN GTP. So this RAN GTP holds your importin beta and recycles back to the cytoplasm, right? After recycling, RAN GTP will undergo hydrolysis. Why? Because you know that you are having the GTPase protein present in the cytoplasm. Meanwhile, you are having importin alpha, right? So they has to be transported back to the cytoplasm. For that purpose, you are having exportin protein. So these exportin protein are responsible for taking your importin alpha back to the cytoplasm. Right? Clear? Is this picture and explanation is clear to everyone? So that's the same thing being mentioned over here. See, statement C is incorrect. Why? Because NTF2 is responsible for the transport or import of RAN within the nucleus, not within the cytosol. Remember this one. Okay. And each importin or exportin mediated transport cycle definitely will remove one RAN GTP molecule, right? With the help of one RAN GTP only, the proteins are being exported and imported. So it is this NTRs. NTRs means nuclear transport receptors. They are responsible for refueling the RAN GTP gradient. What is that RAN GTP gradient? Nuclear transport factor 2. And it is your nuclear transport factor 2 that helps in retrieving the RAN GDP back to the nucleus. Right? Is it clear to everyone or having any doubt? So that's the reason you have got this as the incorrect statement. NTF2 transports RAN GDP into the cytosol it is present. Rather, it has to be nucleus. Right? And this I have shown you the picture. This also I have shown you the picture. This also I have shown you the picture. The export of mRNA is not directly dependent on RAN. Meaning what? See, once the mRNA is being synthesized, it will undergo various processing. Only after the processing and binding of several proteins, it will be transported out. One such protein that is responsible for binding is your NXF1 and NXT1 present in humans. These are the proteins that are responsible for binding to this mRNP protein composition. And after that only, your mRNAs will be transported out of the nucleus. Okay. I hope so this question is clear now to everyone. Yes. So here is the next question to you. The next question to you is, cellular levels can be as high as 100 to 400 micromolar. Of this, unpolymerized actin concentration can be as much as 50 to 200 micromolar. However, the critical concentration for actin polymerization in vitro is around 0.2 micromolar. Some of the following proteins will inhibit polymerization of actin in cells. See, whatever the numbers they have shown over here is just to mislead you. Actually, you have to find out what are these proteins. Among these four options, which protein is responsible for inhibiting the polymerization of actin, or you can say inhibition of actin proteins is catalyzed by which among them. That is the question, but they have just given these numbers just to confuse you. Can you find out the combination of inhibitors that are responsible for actin polymerization? Yes. Was this tough question? Were you able to attend this question, everyone? Yes. Thymosin beta 4, capping protein, that is CAPZ, tropomodulin, and XMAP 215. Have you attended this question or left off? Mostly what happens is that whenever they ask questions from inhibitors, it is very easy to attend. Not only from unit 2, but from any units. But this time it was bit twisted form. It was something very new this time, right? Is there anyone who could attend this question? Yes. Is there anyone who can attend this question? No one? Okay. Sapalta has given the answer. Okay. What about others? Kavita, Devanshi, Akriti. Anyone who has given the answer, who has written it in the exam? Well, the correct combination of inhibitors for actin polymerization is, according to your CSIR answer key, it is 1. Now, what is 1 indicates? It indicates A, B, and C. That means thymosin beta 4, capping protein CAPZ, and tropomodulin. These are the three proteins that are responsible for inhibiting the actin polymerization. Right. Now let's find out how they are able to inhibit the actin polymerization. See, first one is when they are bound to actin, we have got thymosin beta 4. So they will strongly inhibit the exchange of nucleotide binding. If you remember, when there is an actin polymerization, we also see as actin treadmilling. So there will be a continuous exchange of nucleotides taking place. We have got ATP bound form, ADP bound form. So that exchange is being blocked by thymosin beta 4. Coming to the second option, that is capping protein. They are responsible for regulating actin polymerization by binding to the barbed end of an actin filament. Barbed end of an actin filament in the sense plus end. So what happens? It will lead to blockage. Blockage of what? Addition and loss of actin subunits. Third option was tropomodulin. So tropomodulin is a kind of cytoskeletal protein that can bind to the end of erythrocyte tropomyosin, and they are responsible for inhibiting tropomyosin binding to actin. So what is the function of tropomyosin in actin? Anyone knows? What is the function of tropomyosin in actin? Anyone who might have studied in the earlier classes or anywhere from your book? Tropomyosin are actually responsible for binding to the actin for providing stability to the actin filaments. In fact, this tropomyosin as a whole will be bound throughout the length of actin filament in order to provide the stability. Okay. And the last option was XMAP 215. So this is basically a microtubule associated protein which is found in xenopus. Okay, and they are responsible for microtubule growth. So in our question, what they had asked about actin polymerization. But Contradictory to that, XMAP is a microtubule related. That's why we have got ABC as the correct answer. Okay. Next question. To delineate the steps in endoplasmic reticulum transport, a PhD student homogenized pancreatic acinar cells to isolate microsomes. Okay. Now, this retains most of the biochemical properties of the ER. For this experiment, the student has planned a number of controls as mentioned below. Treat one set of microsomes first with detergent and then with protease. Second, treat one set of microsomes with protease only. Third, treat one set of microsomes with micrococcal nuclease. And fourth, treat one set of microsomes with detergent only. So you have to find out the correct combination of controls. Are you able to attend this question? Yes. You have to find out the correct combination of controls for studying endoplasmic reticulum transport. Yes. Find out the correct combination. This question as well as the actin inhibitor question was bit tough to be answered, honestly speaking. But yes, you can try at least this question. Yes, anyone who can answer the question for this? Who can answer this question? What happened? The energy level is very low today. Unit 2 was that much difficult for you to attend in the exam? Yes. Unable to give the answer? Okay, let me show you the correct combination of controls. See, your correct answer is 1. That is A, B and D only. Well, from the option itself, you can clearly determine this thing. Why? Because what is the function of nucleus here? Normally, the function of nucleus is to do something with nucleosomes or with DNA, anything like that we will be having, right? But here we don't have any such thing. Here we are studying about endoplasmic reticulum, and endoplasmic reticulum has no relation with nucleuses. Of course, if we use detergent and proteases, we can get something. But with nucleases, we don't have anything to be taken, right? Because these are specifically needed for DNAs or something related to DNA, right? So let's check out the explanation now. See, microsomes are nothing but a set of vesicles which are formed from endoplasmic reticulum when the cells got degraded, right? Next, and these vesicles can be obtained by differential centrifugation. So upon centrifugation, you can get 3 structural features. What are those features? Rough vesicles, smooth vesicles, as well as ribosomes. Next, microsomes are the most widely used preparation of endoplasmic reticulum membranes, and they are used for co-translational processing of membrane proteins. So this is all about microsomes. Now how this microsomes is related to our question, let's check it out that. What is the function of detergents? Detergents are used to lyse the cells, right? So you might have used it in your labs as well. They are responsible for releasing soluble proteins, membrane proteins, as well as lipids. What is the function of proteases? These are responsible for breaking the peptide bonds present within the proteins. So if you find in control experiments, all the 3 are used. either individually or in combination. That's the reason we had excluded nucleus and we have taken the other three statements. Right? So, micrococcal nucleus, these are commonly used for defining the nucleosome position and chromatin architecture. So, we do not need this for the control experiment. So, that's what I have initially said you. Nucleus means they are related something to DNA. It has nothing to do with your ER proteins, right? So that's why the answer will be your A, B and D. Now, shift 1, this much was the questions. Now moving on to shift 2. The first question to you is, again shift 2 also part B questions were very easy and direct questions. Membrane enclosed organelles often have a characteristic position in the cytosol. So when it comes to animal cells, for example, the Golgi apparatus is located close to the nucleus. Which component is directly involved in ensuring correct Golgi localization in animal cells? You have got actin skeleton, microtubules, nucleolus, peroxisomes. I hope so you might have given the answer for this question. Which component is responsible for proper localization of protein, Golgi within the animal cells? Yes, answer will be Dilish. Okay, I got your answer. What about others? Have you all attended the question? Yes, Safalta. Good response. Others? Madhura, okay, 1. See, you have to find out the component that is responsible for the localization of Golgi complex present within the animal cells. Meaning which proteins are responsible for localizing the Golgi apparatus within the animal cells. Yes, Anaga? Okay, let's find out what is the answer is. The correct answer is 2, that is microtubules. Now what happens here in mammals, we have got a single Golgi apparatus which is usually located near to the nucleus and closer to the centrosome. Okay? And these tubular connections are responsible for linking the stacks together. What is the stacks here? What is the stacks here? Can anyone say? What is the stacks? Stacks means your cis phase is there, trans phase is there, mid phase is there. They are made up of what? Cisternae. So that is known as stacks. So tubular connections will be there among the stacks, right? And localization and tubular connections of the Golgi apparatus, they are dependent on microtubules. Okay. So Golgi microtubules are responsible for contributing the Golgi structure, maintenance of Golgi positioning once the membranes are formed. Yes, Delish. So, in fact, in several experiments, it was proven that microtubules are depolymerized. As the microtubules undergo depolymerization, the Golgi apparatus loses the mutual connections and they will become individual stacks throughout the cytoplasm. Right? So that's why your correct answer is microtubules. Next question to you. Signal sequences that direct proteins to the correct intracellular location. You have to find the correct sequence that is used to import proteins into the nucleus. Now, this is the third question which I come across with respect to nucleus. What will be the answer for this? What will be the correct sequence for this? The correct sequence through which a protein can be imported into the nucleus. So now you learned how a protein is imported into the nucleus. what are the receptors responsible for that, and what are the proteins that helps in the transport, right? Now tell me, what is the sequence that is responsible for the transportation? Yes, very good, Antara. What about others? This is also a very direct question, easily attendable question, in fact. Yes, still did not get the answer, excepting Antara. What about others? You have not attended this question. Yes, very good, Antara. This is actually your NLS sequence, that is nuclear localization signal sequence. Yes, nuclear localization. No, export sequence will be different, Antara. Localization. Import sequence will be different. Export sequence will be different. So this is specifically for import sequence. Okay, very good, Madhura. So the correct answer is 1. So if you simply remember this particular table from Albert's, you would have easily fetched 2 marks. See, import into the nucleus, this is what the sequence. If you follow Lodish also, there what it is mentioned, they are rich with basic residues. So basic residues means what? They are nothing but lysine and arginine, right? For export, they have got a different sequence. If a particular protein has to be imported into the mitochondria, again they will be basic residues, but they are not adjacent to each other, meaning they are distant from each other. But in this case, lysine and arginine are adjacent to each other. That is the difference. Yes, Antara. If a particular protein has to be imported into the plasmid, it must be rich with serine and threonine residues. If a particular protein has to be imported into the peroxisome, then it should have SKL as their sequence. If it has to be imported into the ER, KKXX, if you remember, that is the sequence responsible for the import. If a protein has to be transported back to the ER from the Golgi, then you will have the Kedal sequence for that. So sequences are always an expected question from CSIR. So don't miss on this. Apart from that, we have a small discovery also based upon the sequence. Okay. Simian virus 40. That is a kind of virus having a virus encoded protein known as T antigen. Now, what is the function of this T antigen? They are responsible for viral DNA replication. Okay. So, if they are involved in replication, definitely this T antigen is present where? Present within the nucleus. Okay. So, the signal responsible for this nuclear localization was first identified when they found that there is a small mutation occurred within the lysine residue. So when this mutation occurred within the lysine residue, what happened is that instead of transporting to the nucleus, the T antigen remained within the cytoplasm. Then further studies revealed that this T antigen is composed of a 7 amino acid sequence, that is this one. Right. So from here onwards, a conclusion came that whatever the protein that has to be imported inside the nucleus will have this conserved amino acid sequence. In fact, other proteins that is imported from there also, they deduce this sequence, the NLS sequence. Okay. So that's why first option is the correct answer. Now, next question to you is, following statements were made about protein trafficking in cells. Cargo selection occurs when code proteins binds to the sorting signals either directly or indirectly via adapter complexes. Protein export from the ER is exclusively mediated through COP2 vesicles. Identical code protein is used in both exocytic as well as endocytic pathway. Tethering of vesicles involve GT pieces of RAF family. Clathrin-coated vesicles will transport proteins from plasma membrane to trans-Golgi to late endosomes. Now you have to find the correct combination of statements. From protein trafficking, more or less the questions were very direct. If you know if you have attended the classes or if you have gone through your notes and books, definitely you will be able to attend the question. Specifically from protein trafficking. So can I get a correct answer from your side? Protein trafficking in cells. So you have to be very clear about the COP2 vesicles, COP1 vesicles, clathrin coated pits. Okay, Antara has given the answer. What about others? Others? Madhura, Safalta, Devanshi, Anaga. Yes. Any other who would like to give the answer? A, B, and D. So if you are saying A, B, and D, there should be some issues with C and E, right? So what could be the justification for that? Why C and E are incorrect in that case? Yes? Others? Yes. All of you are trying. As you mentioned, the correct answer for this question is answer 1. Now, what is the explanation for that? Simply, if you remember this table, right? In the question, you have seen that they have asked about COP2 coated vesicles. So, proteins that are exported from ER, from the ER they have mentioned, is carried out by COP2 vesicles. So, from the ER means, yes, Madhura, yes. Yes. From the ER means it is moving to the Golgi. So you have COP2 vesicles. That is correct. Next, cargo selection occurs when coat proteins will bind to the signals directly or indirectly via adapter. Definitely, you should have adapted proteins involved for binding and getting the signals, right? And D, what it is that tethering of vesicles is carried out by GT pieces. We will look into that. The last option over here is clathrin coated vesicles will transport proteins from plasma membrane to trans-Golgi to late endosomes. What is given in the picture? As you can see, clathrin coated proteins are responsible for transportation from trans-Golgi to endosome or plasma membrane to endosome or Golgi to lysosome, melanosome or platelet vesicles. So which is not present in your option. So that's why that is incorrect. COP2, as I said you, ER to cis Golgi. COP1 is just opposite to that, right? Now looking on to the tethering vesicles, see, once a transport vesicles has to be this table is from Lodish Safalta. This table I took it from Lodish. You can refer to Lodish for that chapter 14. And the explanation for the transport vesicles is that, see, we have got a transport vesicle that will be bound with target membrane. So how they will be bound with target proteins? With the help of corresponding snare proteins. Snare proteins means we have got specific snare present within the vesicles and specific snare present within the target. Okay. Apart from this binding, we have some tethering proteins present. So you have got RAB GTP present within the target and you have got RAB GTP present within the vesicle. So they will try to pull towards each other. So end of the day, you will get like this. Right? So that option is also the correct one. So that's why you have got A, B and D as the right answer. Next. Now, the following statements are made with reference to the structure of nucleosome. That is, histone tetramer in the core of nucleosome comprises H2A, H2B, H3 and H4. B. The N-terminal tails of the core histones are believed to stabilize 30 nanometer fiber of nucleosomal DNA by their interaction with adjacent nucleosomes. C. The post-translational modifications occurs within N-terminal as well as globular domains, modulate the transcriptional events. Fourth one, according to the zigzag model of 30 nanometer fiber, the linker DNA circles around the central axis of the fiber as the DNA moves from one nucleosome to the next. Now here also you have to find out the correct combination of statement. Yes. Let's check out who is going to give the correct answer here. What is the correct combination of statements? You can find it over here. Yes, you are most welcome, Safalta. So if you have studied the basic structure with respect to nucleosome, this is a kind of questions where you can eliminate the option and get the answer. So what could be the answer here? Very easy and direct question again. Is there anyone who can give the answer? Yes? No? Okay, let me give you the answer. Yes, I can see Madhura Antara has given the answer. Anyone else would like to have a try? Okay, so the answer for this question will be 1. Now, what is 1? B and C only. Right? That means, is it a tetramer? Do you think so? This is a tetramer which forms between H2A, H2B, H3 and H4. Do you think it's a tetramer? How can that be possible? That is not a tetramer, right? And last one is with respect to zigzag model. So let's find out why B and C is correct option. Okay. So histones H2A, H2B, H3 and H4 are known as the core histones. Right? Yes, exactly, Antara. It is an octamer. And histones H1 and H5 are considered to be the linker histones, right? So these core histones will exist as dimers and the 4 dimers will come together forming an octameric nucleosome core. So obviously you can eliminate the first option, right? Then the linker DNA that circles around the central axis as the DNA moves from one nucleosome to the next occurs in solenoid model. If you remember, we have got 2 models for their explanation. That is, one is solenoid model, another is zigzag model. So this is an explanation of solenoid model for that. And if you look into the Watson book, you will find that directly the sentence is given over there. Right? Then explanation for the other 2. See, this is the structure of 4 different histones. We have got N-terminal region and histone fold domains. And in the statement, what it was mentioned, these are the 2 regions which can undergo frequent modifications, right? So majority of modifications occurs in the tail regions, that is in the N-terminal region. More precisely speaking, it is your H3 and H4 which undergoes frequent modifications. Of course, some modifications will also occur within the histone fold, which is known as your globular domains, as you can see in this picture. If you see in case of H3 and H4, you can find the lysine residue is methylated over here at 79th position, where within the histone fold domain. Similarly, in H4 also, lysine residue is methylated, where in the 59th position. So that's why that statement is also the correct statement. Okay. So that's all from my side. I hope so. You got some information about where you went correct, where you have to work on yourself in the upcoming exams. So, wishing you all the best for that. Now, I am passing my controls to Dr. Violet, who will be discussing with you the questions from cell cycle as well as microbial physiology. Thank you so much, everyone.
Good evening, everyone. Am I audible? Please confirm if I'm loud and clear and you all are able to see the slides as well. Good evening, Antara. Good evening to all of you. Yes. So are the slides visible? Okay. Okay. So thank you, Divya ma'am, for giving such a detailed explanation about the various questions that were asked from Unit 2. So I'm sure you all have, understood like why a particular statement is right, why it is wrong, and looking further into it. So here I'll be discussing the questions that have been asked from Unit 2D and 2E as well. Okay. So before I start, what did you all feel about the questions that were asked from Unit 2D and 2E? Were it easy? Were it difficult? Yes, Madhura, thank you for confirming. Tell me, what do you think about the questions that were asked? So I can, get an idea. What do you all think about the questions from Unit 2D and 2E? Easy? Okay, good. Right, some of the questions, now if you see from the part B, okay, so we'll explore those questions and we'll find out. Some were easy, but yes, 2 or 3 were difficult. Okay, so like some questions from shift 1 as well as from shift 2. So let's start with our discussion. So first question was from part C. So I have gone ID wise. Okay, so this is a part C question that was asked in shift 1. Following statements were made about chromosome cohesion during mitosis and meiosis. Okay, so these are statement based questions. You have to find out which one are the correct statements. So let's see the statement first. Though cohesion is important for pairwise alignment of the chromosomes on the mitotic spindle, it is not important for the generation of tension across the centromere. So this is statement A. Statement B says cohesion binds to the chromosome even before the initiation of S phase. Madhura, it is shift 1. Okay. Then, third statement in Fission East, centromere specific localization of MOA1 and REC8 regulates the orientation of kinetochores at meiosis 1. D. Cohesion exhibits uniform distribution or localization pattern across the chromosomal length. E. Polo CDC5 is a positive regulator of separase and endopeptidase that facilitates opening of the cohesin ring. So, what do you all think is the correct statement over here? So, just try and tell me. So, these words, these terms is something that is very common general terms. Okay, we know about cohesion, where it is present, we know what is centromere. Okay, so it is not that difficult, I guess. So just try and tell me what should be the correct answer over here. Okay, very good, Antara. Yes. So that is why you said it is easy question. Yes, right. What about others? Anyone else trying? No. Unit 2D is very important from CSR point of view. So you can see if you say it is easy and you have got the right answer, so easily you fetch around 4 marks, right? Okay, so let's understand this question over here. Let's see in more details. So the first, okay, so the right answer, as some of you all have said, the right answer is your option 4. Okay, so now option 4 is B, C, and E, right? So what it says, the first statement, cohesion is important for pairwise alignment of the chromosomes on the mitotic spindle. Okay, so definitely it is responsible for pairwise alignment, but it is also mentioned it is not important for generation of tension. Is this correct? No, right? It is important for the generation of tension, right? Because of the presence of, if you see the presence of the cohesins, so these are only responsible, so this is only responsible for the pulling of the cystochromatids, right? If it is not present, so there will be no proper separation of these cystochromatids, right? So that is why the statement A is incorrect. Clear? So cohesion is important for generation of tension across centromeres. It counteracts the pulling force of spindle microtubules, which ensures the bipolar attachment of chromosomes. Right? So that is why statement A is incorrect. Now, statement B says cohesins bind to chromosome even before the initiation of S phase. Right? So, yes, B is correct because cohesins associate with chromosome during G1 phase only, right? Before the S phase. But yes, what happens? They acquire their cohesive properties, that is, the properties to hold the cystochromatids during DNA replication. So, that is why B is correct. Now, the statement C, what it says in fission yeast, centromere specific localization of MOA1. What is it? It is a protein that is monopolar attachment protein 1 and REC-E. So, it regulates the orientation of kinetochores at meiosis 1. Right? So, what this protein does, MOA1, it is responsible for chromosome segregation during meiosis 1, allowing meiotic rec 8 to establish centromeric central core. So this is one of the important protein, right, that is responsible for chromosome segregation. And rec 8, it binds the cystochromatid. So that is why over here we find that statement C is again correct. So in fission, the fission yeast meiosis specific protein MOA1 interacts with REC8 and localizes to the core centromere. So it is responsible or required for establishing the orientation of kinetochores. So kinetochores are oriented at one side, right? It is not that everywhere they are present like this, no, right? So this orientation is possible because of the association of these proteins, right? Then cohesion exhibits uniform distribution localization pattern across the chromosomal length. What do we see? It is not uniform distribution, right? So that is why D is incorrect over here. Why D is incorrect? Because if you study in details, you'll find that cohesion does not exhibit uniform distribution pattern across the chromosomal length. Okay, so what is happening over here? You'll find cohesin along the chromosome arms is spaced at lower density at intergenic regions. What are intergenic regions? Between the genes, the sequences that are present. So there, the cohesins are not present, or they are at lower density. So they are not uniform. So that is why statement D is incorrect. Clear? Then coming to the statement E, where polo or CDC5 is a positive regulator of separase. Now, separase is very important protein. Why? Because what it does, it helps in cleaving. Okay, so this separase At the onset of anaphase, it is endopeptidase called separase. This is activated to cleave the subunit SCC1 and RAD21. So this SCC1 is cystochromatic cohesion proteins. So only when the cohesion rings are cleaved with the help of separase, so that will only help in chromosome segregation. So that is why statement E is correct. Clear? Everybody understood the various statements? Right? Okay. So I'm sure you'll have noted down all the important points. So these points will help you when you are going for the next exam. Okay, now let's see the next question. So again, this is from shift 1. During cytokinesis, a small GTPase, rho A1, localizes to the equatorial membrane above the spindle midzone. The localization activity of rho A is potentially modulated by, and there are various proteins, okay, names are given. So you have to find which ones are the correct ones over here. Okay, which combination is correct. So rho GEF ECT, ECT2, B is Aurora B kinase, C is PLK1 kinase, D is MKLP1 kinesin, E is ATM and ATR. So which of the combinations are correct? Tell me. So which proteins are responsible during cytokinesis? Try and tell me. So this is a little difficult question, I will say. Okay. But if you have an idea about some of the proteins, like if you know GEF, what it does, Aurora B kinase, okay, polo-like kinase. So these are something that we study. Okay. So if you know at least these two, so you can easily find the right answer, because if you know at least B and C, so you will get an idea, like B and C should be there in the option. So in that way, you can easily get the right answer, right? So the right answer, anybody is trying? No? Anybody answered this question? Try and tell me. So when I said B and C, I said, right, it is there in the correct statement. So what do you see? Which option should be correct? Madhura, will it be 4? Okay. Yes. So the right answer is going to be option 2. Clear? So what I said, B and C. So where you find B and C, in which option? Okay, so this is how, these complicated questions, if you know at least two of them, so you can easily get the right answer, right? Now it is not just B and C, so there are other proteins also that are involved over here. So I'll give you a detailed explanation over here. You all can make a note of it and try to remember these details. So whenever we talk about animal cells, so when during cytokinesis, the furrow that is formed requires activation of GTPase rho A. Okay, how it gets activated? With the help of guanine nucleotide exchange factor, that is ECT2. Okay, so that is why rho GEF ECT2 is one of the options. Okay, so that is why you see over here that A is correct. Okay, so all these proteins are there except for ATM and ATR. Okay, so first one, you got an idea what it does. Then Aurora B. Okay, again, it is a very important protein. It phosphorylates MGC RAG gap. So what it does, it phosphorylates this protein and then it can activate this gap rho A. Okay, and this phosphorylation is important for restricting rho A activity during furrow ingression. So it is required during furrow ingression or terminating rho A activity. So it is like a regulating protein, the Aurora B. Okay. Then next important protein is polo-like kinase 1. What it does, it controls the initiation of cytokinesis through rho GEF protein ECT2, through this protein. So ECT2, for that you need polo-like kinase 1. Okay, the initiation of cytokinesis. What is cytokinesis? The furrow formation, the division of a parent cell into two daughter cells, right? So it controls the initiation of cytokinesis through the Rho-GEF protein ECT2 and the GTPase Rho-A. Okay, as well as another important function of polo-like kinase, it'll help in exiting the cytokinesis. Okay, so that is why again polo-like kinase will play an important role, right? Then D, cytokinesis requires activation of GTPase Rho-A ECT2 exchange factor responsible for Rho activation. So all this we saw. And then finally, there is a central spindlin. Okay, so it is composed of the Rho family GTPase activating protein Rho gap, MGC RAG gap, CYK4, and the kinesin. So the kinesin that is mentioned in question. So all these proteins are further helping in activating ECT2. Okay, now this is one of the question that they have given from a research paper. Okay, so you see that you have to even go through the recent research paper and try to find out what are the various proteins that are involved in mitosis, meiosis. Okay, and then the last one, so we saw A, B, C, D, all these four options are correct. Now what about ATM and ATR? Now if you have studied ATM and ATR, I'm sure you'll have studied, so we know it is not involved in furrow formation, right? So it will activated by DNA damaged, right? So that is why during DNA damage and DNA replication these proteins are getting activated. So this is not a part of your answer. So easy 8 is not the E option is not involved, ATM and ATRs, right? Other proteins are involved. I'm sure you'll have taken a note of it. Yes, Madhura, this question, as I said, it is a part of a research paper, recent research paper. Okay, you'll get all these proteins in details. Okay, so should we move ahead now? I'm sure you'll have got an idea about the various proteins. And now next question is again from part C from shift 1. DNA replication occurs in S phase. The entry of cells into S phase is regulated by the tumor suppressor protein RB. The statements below are made with reference to the role of RB. Statement A, RB binds to E2F in the cytosol and prevents E2F entry into the nucleus. B, RB is phosphorylated by cyclin A, CDK4. C, phospho-RB activates E2F. D, E2F activates cyclin E production, which promotes the G1 by S transition. Which one of the following options represents all correct statements? So this was a pretty easy question. You all might have an idea about it. Okay, Madhura. So just try and tell me. I'm sure you all have, the students from shift 1 have answered this question. Quite easy. So what do you all think? What is the right answer over here? Think and tell about. You all have studied about RB, I'm sure. What should be the right answer? You have to find the correct answer from here. So which all statements you all think is correct? Difficult? Not able to solve? Okay. So let's find it out if how the question has to be solved. So for this particular question, what I would say and suggest is you have to remember this figure, right? So if you see this figure, what you see over here is RB protein holds the E2F transcription factor, right? So when RB protein gets phosphorylated, so RB becomes inactivated. Then the E2F becomes active. Okay, and it leads to further transcription and translation, and that leads to cell proliferation. Now, what is the right answer for this question? Question number 88. The right answer is your option 3. Okay, why option 3? So we saw the figure, and now let's try and understand how to solve. RB binds to E2F in the cytosol and prevents E2F entry into the nucleus. So that is what we saw. Okay, RB binds to E2F, and then E2F is a transcription factor. So when RB is bound to E2F, E2F cannot move inside the nucleus, right? So that is why option 1 is correct. Then RB is phosphorylated by cyclin ACDK4. So over here, if you see which proteins phosphorylate RB, okay, so RB is actually phosphorylated by CDK4 cyclin D complexes. Okay, so that is why the second option is incorrect. B is incorrect. Then phospho RB activates E2F. So that is what we saw when RB becomes phosphorylated, E2F becomes active. So we can say phospho RB activates E2F. So C is correct. Now D, E2F activates cyclin E production, which promotes the G1 to S transition, right? So definitely yes. So this is over here, if you see, so it leads to the transcription of various proteins. So which proteins are those? So those are your cyclin E. Okay, it encodes the gene that codes for cyclin E. E2F stimulates expression of its target genes, which encode proteins required for cell cycle progression in G1, including the G encoding cyclin E. So that is why statement D is also correct. So what is incorrect is option B. So A, C, D is the right answer over here. Okay. Now let's move on to the next question. So you have to just remember this figure. So it will give you an idea about how to solve the question. So before I move further, so seeing the standard of the questions, if you are feeling you need more preparation, so definitely join the Shaurya batch. Okay, and today the Shaurya batch is starting, so still you have time, though the seats are almost full, but for night it will be open, so you can join this batch, and furthermore you can get the right direction for preparation. So the classes will be very unique. So these are known as raid classes by CSIR Net Experts. You will get interactive online classes, 24/7 doubt solving via chat system. Then even you will get access to the printed hard copy study material, as well as you will get access to the video backups of all classes and much more. So if you feel you need more preparation, do call at this toll free number that is shown over here. Okay, now coming to the next question, part C question from shift 1. The table given below lists types of extremophiles and the environments that they are adapted to. Which one of the following options represents the correct match of the columns? Okay, so you have list 1, that is the type of extremophile, and list 2, that is the environment. So this is a very simple question. It is part C. If you know this question, you easily get 4 marks. So just try and tell me what should be the right answer over here. I'm sure you all know this. You will get the PDF, Sunil. I'll talk about it at the end of the session. So stay tuned. I'll tell you how you can get it. Yes. So tell me, what is the right answer over here? What do you all think? Just the terms you have to match. Very simple question. Psychrophile, what it is? Okay. So let's see the right answer over here. So the right answer for this particular question, that is question ID 120, will be option 3. Okay, so here just you have to know these terms. So if you have studied unit 2E, this is question from E to E, psychrophile. Okay, psychrophile means those organisms that grow in very extreme cold temperatures. Okay, so you have to match A with 5. Yes, Zishan, correct, you are absolutely right. Yes, good. Now, hyperthermophile, by the term itself, hyperthermophile means the ones that are growing at very extreme high temperatures, right? So B you match with 4. Okay, so if you know B4, so you can see easily, there is no other option where B4 is matched. Okay, so you get the option 3. Then alkaliphile. So what it is? So alkaliphile means high alkaline environment. So those organisms that grow in very high alkaline environment, we call them as alkaliphile. Hyperpiezophile. Hyperpiezophile are those organisms that are present in very high pressures. Okay, and halophile is something very common, the ones that grow in very high salt conditions or high salinity. So the right answer is your option 3 over here. Yes, Devanshi, good. Very good. Okay, so the terms and their matching is written over here. So we move on to the next question. Question ID 139. It is again a part C question from shift 1. Now again, this was a very simple question, but what I found in this particular question was there is an error in the question. Okay, so even if you have solved it, so you'll get the benefit of doubt. So the question ID 139 says a culture was started by inoculating the medium with 100 cells having a generation time of 2 hours. Assuming the culture grows in ideal synchrony for at least 24 hours, what will be the number of cells in the culture at 2 hours and 9 hours? So tell me for this question, which formula you should use? Fast. I'm sure those who all have attended this shift 1, you all know the formula. So which formula should we apply for this question? What is given? Okay, a culture was started by inoculating the medium with 100 cells, means it is N0, it is given. Okay, and time is given. Having a generation time is G is given 2 hours, and total time is given, like for one case it is 2 hour, and for another case it is 9 hours. Okay, but over here, if you take 9 hours, you will not get the correct answer. Okay, so what I have found, if it is 8 hour, so you will get the right answer, which is there in CSIR, that is option 1. Okay, but if you have solved this and you have at least no chosen any of the options, so you will get the benefit of doubt. So let's see how to solve this question. N0 is given, G is given, T is given. So you have to find what will be the number of cells, means you have to find NT. So what is the formula? The formula is NT is equal to N0 into 2 to the power N. Okay, where N is the number of generations. Okay, so I'll just explain this formula. NT is the total number of cells after a particular time t. Okay, and N0 is the initial number of cells. Okay, initial count, you can say, that is 100 that is given. And NT is equal to N0 into 2 to the power N. So how will you calculate N? N is time by generation time. Okay, like for first case T was 2, so 2 by 2 it becomes 1, so N becomes 1. Now when you use that in the formula, Nt is equal to N0 into 2 to the power N, where N is 1, so you'll get 2 into 10 to the power 2. Okay, so this is first case. Now in the second case, because they have given 9 hours, so if you use 9 hour and try to find out, so now the answer will be like, you will get N by 2, that is 9 by 2, you'll get around 4.5. So NT becomes 100 into 2 to the power 4.5. So it is something around this value, 2.26 into 10 to the power 4. Okay, but if you take 8 over here and then you try to do the same question, you will get option 1 as the right answer. Okay, so what is important? If you know the formula, it is a very simple question. You can easily solve the question even if it is not matching, and you have selected and you know that your answer is correct, and you have selected a particular option, the nearest one that you see over here, maybe this one or first one or second one, so you will get the benefit of doubt. Okay, and you will get 4 marks. So it was a simple question. Okay, clear for everyone? Should I move ahead? Can I see a BIO if everybody is understanding everything? Can I see a BIO? If you type BIO, I will move ahead for the next question. Okay, Madhura, yes. So I see BIO coming up. So let's move ahead. Okay, so now we come to shift 2. So it is a part B question from shift 2. A mutation in which one of the following sigma factors may be a possible cause for E. coli failing to adapt in response to thermal stress. Now, for thermal stress, now there are various sigma factors. Okay, so they have given the various sigma factors, and you have to find out which is the correct answer, like which sigma factor is responsible for adaptation in response to thermal stress. Okay, now over here, I will tell you the right answer. This was a very simple question. I'm sure many of you all might have got it right. So the right answer over here will be option 2, that is your sigma 32. Okay, sigma 32, remember this sigma 32. What is this sigma 32? Okay, this is the protein that actually helps in, these are subunits of the bacterial RNA polymerases. So these are proteins responsible for determining the specificity of promoter DNA binding and control how efficiently RNA synthesis is initiated. Means there'll be expression of that particular gene. So each sigma factor is connected to a particular gene expression. Okay, so sigma 32, if you see, it is required for normal expression of heat shock genes. Okay, so if mutation in this sigma 32 occurs, so it will be unable to adapt in response to thermal stress, right? Then coming to, so the right answer is your sigma 32. Then what about the other sigma factor like sigma 70? Okay, it, this acts not only in initiation, so it is responsible for initiation as well as in an early stage of elongation, so to induce a transcription pause and simultaneously to allow the phage lambda gene q transcription anti terminator to act. Okay, so that is the role of sigma 70. Then there are many sigma 54 dependent operons, and they are involved in nitrogen assimilation and metabolism. So these are the important sigma factors you, and the use of these sigma factors you should know. Okay, so you can note it down for your further reference. Okay, now coming to the next question, that is question 222. It is from shift 2. Following statements were made about stress response in prokaryotes. Okay, so you see a lot of question is asked from stress response. Okay, so many students, they tend to quit this part, the stress response. Okay, but you see that the questions are not very difficult, and if you have studied at least once the topic stress response, so you can easily solve the questions. Now let's see this particular question. Per functions as a major peroxide sensor in many gram positive bacteria. Extreme acidic pH induces REC mediated DNA damage, which in turn may induce virulence gene expression in some pathogenic bacteria. C. Induced expression of heat shock proteins neither protects the cells from heat nor plays a role in bacterial virulence. D. In gram negative bacteria, heat shock sigma factor regulates the transcription of the major heat shock proteins. E. GRO-ES is an ATP-dependent chaperonin, but GRO-EL may function in ATP-independent manner. So you have to find out which is the correct combination of the above statements over here. Okay. So now for this particular question, if anybody has tried, so we'll see that first thing, okay, like what we saw over here in the previous question, we saw that sigma factors play a important role, right? So what we can say, option D should be included in the answer, right? So we see that two options like these three has D. Okay, so D, what we can say, is a correct option. Clear, right? And then per-R functions as a major peroxide sensor in many gram-positive bacteria. So yes, per-R is responsible in many gram-positive bacteria. It is a major peroxide sensor. What does a peroxide sensor do? Peroxide sensor can sense the hydrogen peroxide that is present. Okay, so whenever it can sense in the bacteria, like the hydrogen peroxide will act as a stress for the pathogen, right, for the bacteria in the gram positive bacteria. So this per R, it can sense the hydrogen peroxide and will activate the antioxidant mechanism in the bacteria. So that is why the option A will be again correct. Clear? Now B, extreme acidic pH induces REC mediated DNA damage, which in turn may induce virulence gene expression in some pathogenic bacteria. So again, this option, okay, is correct. So what we see in various macrophages, so inside the macrophages, if they taken the pathogen, so there is extreme acidic pH, right? So in that case, it will induce REC mediated DNA damage. Okay, so this will lead to the expression of virulence gene in the pathogen. So again, this is a correct statement. So A is correct, B is correct, and D is also correct. So what we see over here, if A, B, and D is correct till now, if you see, so you will find that the option 2 is the right answer over here. Okay, now why C and E is incorrect? So, induced expression of heat shock proteins neither protects the cell from heat nor plays a role in bacterial virulence. Is it correct? No, right? If we say heat shock proteins are responsible in protecting the pathogen against the heat shock, so how come this statement is correct? Okay, this is incorrect because it does protect. Okay, it is saying it neither protects. It does protect the cell from heat when the expression of heat shock proteins occur through the sigma factors. So C is incorrect, right? Now, what about E? So GROES is an ATP dependent chaperonin. So this statement, if you see, one, it's saying it is ATP dependent. Okay, GROEL, it is saying ATP independent, while both GROEL and GROES chaperonin complex is a bacterial protein folding system. So both of it function in an ATP dependent manner. Okay, so what we found out, that this statement is incorrect. E is incorrect. So the right answer is your option 2. Okay, and other statements I have already explained you about per R, about the sigma 32 acidification of macrophage phagosomes provides an essential signal for activation of which pathogen. Basically, if you see, it is salmonella virulence genes get activated. So these genes are required for intracellular survival and growth, right? So now we have understood this question. Now coming to the last question. Following statements were made about cell cycle regulation. Okay, so you have to find the combination of the correct statements. A, de novo synthesis and destruction of cyclin B are essential for cell cycle progression in yeast. B, de novo synthesis and destruction of cyclin B and cyclin dependent kinase are essential for cell cycle progression. C, CDK activity is regulated by both activating and inhibitory phosphorylation. D, retinoblastoma functions as an inhibitor of G2 to M transition. Inactivation of SIC1 is essential for transition into S phase. So, for this particular question, some of them you have already seen. Okay, what we saw, first thing, okay, I'll just show you one figure over here. So, what we find in yeast cell cycle progression, so there is one CDK and different cyclins is required. So when there is one CDK and different cyclins, so what we can say over here is option A correct? Right. So you have to find the correct statements. De novo synthesis and destruction of cyclin B are essential for cell cycle progression. So cyclins and CDKs are important, but there is one CDK and different cyclins. Okay, and those cyclins over here is the cyclin B that is mentioned over here. Okay, so that is why option A is correct. Now option B, what it says, option B says de novo synthesis and destruction of cyclin B and related cyclin dependent kinase. So each the cyclin differs, okay, but the CDK remains the same, right? So that is why option B is incorrect. So it does not have related. Okay, there is just one, as I have mentioned, one CDK along with different cyclins is required. And progression through the fission E cell cycle requires phosphorylation and dephosphorylation of CDC2 residue. So statement A is correct while B is incorrect. Okay, and in this figure, what you see that the cyclins and CDKs. Okay, now this cyclins and CDKs will activate this protein, okay, the protein that are responsible for the fission that occurs in yeast. Okay, now here the CDK has two sites, okay, where it can be phosphorylated. Now one is a activating phosphate that is added and another one is an inhibitory phosphate. Okay, so both activating and inhibitory phosphate can be added. So if you see over here, mitotic CDK along with M cyclin, okay, so this complex of protein here, when V1 is present, so it is inhibitory kinase, it will add the inhibitory phosphate. But when CDC25 is present, so it is a phosphatase, so what it will do, it will remove this inhibitory phosphate. So it will give the active MCDK. Okay, so the next statement, CDK activity is regulated by both activating and inhibitory phosphorylation. So through the figure, we understood that option C is correct. Right? Then D, retinoblastoma B functions as the inhibitor of G2 to M transition. Okay, so now in this case, so RB we saw, right? RB, what it does, it is inhibitor, right? It will hold the E2F, right? So in this case, but what is mentioned over here is retinoblastoma functions as an inhibitor of G2 to M transition, but it is an inhibitor, but it is not responsible for this transition, not for G2 to M. Okay, so what it is responsible for, G1 to S phase of cell cycle, it stops. So that is why this option, that is D, is incorrect. Okay, and now coming to E, inactivation of SIC1 is essential for transition into S phase. Yes, SIC1 is actually a inhibitor. It inhibits progression from G1 to S phase in the cell cycle. Hence, this has to be inactivated so that the cells can enter into the S phase. So now what we find is the right answer over here, because E is correct, the right answer is your A, C, and E. Okay, so now we come to the end of this discussion of questions and answers from unit 2. Now before I leave, I would mention that you please take a note that the PDF of sample question papers along with answer discussion is available in our Telegram group. So you need to join the Telegram group to avail this facility. You can download the PDF from there. Thank you, everyone. So I would say practice like you have never won, play like you have never lost, as mentioned by Michael Jordan, and wish you all the best. Thank you, everyone.